Hi everyone, I'm Mirelijn. I'm doing my PhD at the Amsterdam University Medical Centers with Mark as my supervisor. And today I'd like to tell you something about how codata take omics to the next level. So the motivation for this work is clinical research on omics, and it comes from, in particular, cancer genomics. Some goals in cancer research are early diagnosis and personalized treatment. So research questions are all about, can we predict whether someone has cancer or not, or which treatment will work, or can we predict the risk of when a tumor will come back? So these questions are about prediction, but also on covariate selection, because usually in the first phase of clinical research, they measure the entire genome, but then for follow-up studies, they have to limit themselves due to budget reasons to a small-sized selection of covariates. In this talk, I have an application as example with the following clinical research questions. How well can we predict treatment response with microRNA expression? And which microRNAs should be selected for a small size predictive panel. My PhD is all about learning from codata, and codata is auxiliary information on the covariates. So in this example, we have the covariates microRNAs. Now, codata can come in all different shapes and types, and some examples in cancer genomics are the following four. So we have chromosomes, which are non-overlapping groups of genes on the same chromosome. You may have domain knowledge. So in biological domain knowledge, you have the concept of pathways and gene ontology. So then you have groups of genes that may be overlapping or structured. And you may have results from previous studies from which you can derive summary statistics like continuous p-values. Now, how does codata take omics to the next level? In two senses. Figuratively, we can learn from codata to improve prediction performance and the covariate selection. But also literally, we can use codata to quantify predictive importance not only on the covariate level, but also on the next level, on group level and on codata source level. So I'll mostly focus on the second point, and in the end, I'll briefly touch upon the first point. Now, why is it interesting to quantify predictive importance on a higher level? Let's consider these two extreme cases. So we have some disease that we are researching. There are some mechanisms driving this disease. And on the one hand, we have the case that there are a few covariates that have a large effect. And on the other hand, on the other extreme scenario, we have the scenario in which all the covariates have an effect, but the effects are really small. And cancer and other complex diseases or lifestyle diseases are thought to be the result of many small effects. So more like the right picture. And in that case, you can ask yourself, okay, if all these effect sizes are really small, then if you look at covariate level, one covariate on itself doesn't have a large effect. But if you look at a group of covariates, then a group of covariates can have a large effect. So it might be more interesting to compare predictive strength of groups of covariates. Now, this is where the codata comes in. We use the codata to define these groups, and we can have multiple codata sources. And both groups can differ in predictive strength, but also codata sources may differ in predictive strength. So as an example, if I have these many small effects and I might have two groupings, a green grouping and a blue grouping. And in the first blue grouping, you see that for the first group, there is a difference in average effect size in the first group and in the second group. But in the second grouping and the green grouping, the average effect size is the same in both groups, so that's not really informative. So we would like to quantify the average effect size or the importance of groups, but also the importance of codata groupings. That leads us to our following model. We have a generalized linear model for the response or a Cox survival. So we have the main data X, and we have the response Y, and we have a vector of regression coefficients beta. And we have a link function, so the software currently allows for linear regression, logistic regression, and cog survival. And then we impose normal prior on the regression coefficients on beta, which correspond to a ridge penalty. Now, usually you have one overall penalty, which corresponds to this global prior variance, tau global, but we have a weighted prior variance, so we weight it with a linear combination of weights over codata sources, which obtain a codata weight W, and over different groups, and the group weights are in gamma, and Z is the codata matrix, which codes up the information for each covariate. We use empirical-based techniques to estimate the prior parameter, so tau global, W, and gamma. And because we have this normal prior, we have a nice interpretation of these prior estimates. So let's take a look at that. And the estimates quantify the a priori expected effect size globally in groups and in groupings. So in our empirical base approach, we first estimate the global level. So you can show, because we have this normal prior, that tau global is approximately equal to the average effect size over all the covariates, all the p covariates, times some constant. So that's the same as this dashed line here, the black dashed line. And then in the second step, given this global level, we estimate for each codata set separately all the group weights, and you can show that the square root of the group weight is approximately equal to the average effect size in one group divided by the overall level. So we have the horizontal line in one group divided by the black dashed line as reference. And that quantifies how important the group is. And then if we have done that per codata source separately, then we combine the codata sources in the last step, in which we quantify the contribution of each codata in the codata weight. Now, back to the microRNA application, we want to predict treatment response. So either a patient clinically benefits from a treatment or not. We have data with 88 patients or samples with a bit more than 2,000 microRNAs, and we have 5 codata sets. Before standardizing, we can measure the mean or abundance and the standard deviation. So we have 2 codata sources based on those 2 statistics. And we have 3 codata sources based on fast discovery rates. One is simple 2 groups, a group with tumor-specific microRNAs and one group with the rest. And then we have 2 different fast discovery rates, one for the comparison between metastatic tumor and normal tissue, and one for primary tumor and normal tissue. So we use these 5 codata sets, but we have this continuous FDRs, fast discovery rates, so we have to do something about that. So I'll just briefly explain how we handle continuous codata. The method takes shrinkage to the next level as well. So you can use any type of shrinkage on the group level. And in this case, we use a hierarchical lasso to select groups on group level, such that we can use an adaptive hierarchical discretization. So I have some figures here illustrating it, but the idea is that we have a continuous fast discovery rate, and we suspect that covariates with a low FDR are probably more important for the prediction, but we don't know how low is low, and is that cutoff really clear or is it more smooth? So what we do is that we let the data decide how we discretize the data. So we have these groups, and then we let data decide which groups are selected. And these groups correspond to a discretization. So in this case, we fit the model, and then the gray groups are selected. So you see that up till group 10 is selected, and 10 corresponds to the group with the smallest FDR values. Now, let's go to the results on the question, how important are groups and codata groupings? So we can interpret these prior estimates. On the left, you see the grouping weights, and on the right, the group weights for different folds in cross-validation. And if we first look on the picture on the left, then you see that the grouping FDR2 obtains on average the largest grouping weight, meaning that this grouping is the most informative. And in particular, it is more informative than FDR1. So that suggests that the comparison between primary tumor and normal is more important for the prediction of treatment response than the comparison between metastatic and normal. So that's quite interesting. And if you look at the prior variance weights inside this grouping, then you can interpret these results. So the group with the smallest FDR values obtains the largest prior variance weight. And it means that a covariate in this group has an effect size that is on average the square root of 75 times as large as the global average effect size. And then lastly, what is the added value of codata to prediction and covariate selection? So I compared several models in dense and sparse settings. I have here ordinary rich, which does not use codata, and our method is called ecpc. So you see that you gain almost 0.1 in terms of performance in area under the curve. And in the sparse setting, I compared to elastic net as benchmark. So you see that the improvement is even larger in this case. So if the codata is informative, which it is in this case, then you can gain quite a lot in performance. And we also show that the covariate selection is more stable in the main data and that it validated better on a related outcome on an independent data set on colon cancer in the Cancer Genome Atlas. Now, if you want more information, then you can go to the archive paper or ask me. And there's an R package available on my GitHub account. So if you want to try it out yourself, it's easy peasy. Thank you.